Everyone, my name is Christianah Kehinde. I'm a PhD student at the Division of Computational Biology Division, Department of Integrative Biomedical Sciences, Faculty of Earth Sciences at the University of Cape Town. I'm being supervised by Dr. Osimbenda. I'll be presenting to you today on the topic dual effect of the tumor microenvironment on a newly identified subtype of colon cancer. As a way of introduction, colon cancer is a highly heterogeneous disease and it's marked by substantial intratumor and intertumor variability. Investigating transcriptomic profile can offer insight into this heterogeneity. This had actually been shown in recent reports. However, most genome transcriptomic studies on colon cancer have primarily focused on examining primary tumors and matched normal tissues, often neglecting the multi-stage progression from early to late stages of the disease. Therefore, our research aims to establish unique molecular colon subtypes based on the progression in transcriptomic profile, and would also investigate the implicating factors such as mutation and tumor microenvironment affecting the progression of colon cancer and the implication in diagnosis and therapy. As outlined in Figure 1, in the poster design, the following steps were taken in this research analysis. RNA sequencing data from 39 colon cancer patients were obtained from the XENA database. Then we applied an in-house novel normalization method to capture the heterogeneity in the early to late stage colon cancer development. Hierarchical clustering was used to review the colon cancer subtypes with varying progression and to differentially expressed genes between these subtypes were identified with LIMNA. The differentially expressed genes were then subjected to recursive feature elimination and mutational analysis to reveal driver genes. The tumor microenvironment was also assessed using the EPIC2, and the biological pathways were analyzed using the cluster profile hard packages. Finally, we validated this procedure, this step, with an independent database to validate our study. Analyzing the normalized gene expression profile significantly advanced our understanding of the variability in gene expression that is related to colon cancer progression by uncovering a clear progression pattern from the early to the late stage of the disease. Since we identified two distinct colon cancer subtypes that exhibit differing gene expression profile, we consider the L subtypes the predominant form of colon cancer based on sample size, and the S subtype exhibits substantial divergence from L throughout disease progression. This distinction is strongly supported by the large number of the differentially expressed gene which was identified between the two subtypes, totaling 1856. This was actually gotten after we've applied the stringent LFC threshold. Furthermore, the 16 gene subsets that were selected from the DEG by the RFE method achieved clear segregation of the two subtypes with 100% accuracy, as seen in Figure 2 and 3, the PC and the volcano plot. This result highlights the efficiency of the RFE approach in rapidly identifying a minimal yet highly descriptive gene subset, further reinforcing the presence of substantial molecular heterogeneity between the two subtypes. When we applied this to the independent validation data set, our in-house normalization method also successfully identified two distinct clusters, a large and a small cluster, supporting our finding. The box plot, as seen in Figure 7.3, illustrates 6 of the RFE genes that were found across both the XENA and the GEO dataset. Subtype displayed a stable disease progression from early to late stage with mean normalized gene expression value remaining around 0. In contrast, the S subtype exhibited a statistically significant downregulation of this gene, which suggests a more dynamic increase in molecular activity. Similarly, immune-related genes showed a stable normalized expression around a mean value of 0 in 1, while displaying regulation in heads, further supporting the notion of greater molecular dynamism in subtype heads compared to head. That is in figure 7.2. A key finding in this study is shown in figure 6, and that is the concurrent downregulation of both oncogenes and tumor suppressor genes in subtype heads, which may be driven by likely random silencing mutation or regulatory mechanism within the tumor environment. Investigating the mutational landscape of the most frequently mutated genes, including known driver genes in colon cancer, reveal the potential involvement of additional factors in the round regulation of those genes. For instance, APC is a well-known, tumor suppressing gene, and it exhibited a mutational frequency of 75% and 83% in the small and large subtype respectively. That is in figure 7.1. This difference would potentially imply greater than regulation of APC in hair due to its higher mutation frequency. However, this is not the case, as APC downregulation is more pronounced in the hair subtype, indicating a possible role of TME in suppressing the tumor suppressing genes expression. Interestingly, a negative correlation was observed between the tumor microenvironment fraction of CD4 and CD8 T cells and the differentially expressed genes, which include the tumor suppressing genes and the oncogenes. And both immune cell types manifested increased infiltration as the S subtype progressed, while the gene expression showed a concurrent downregulation. This inverse relationship indicates the possible role of CD4 and CD8 T cells in driving the observed downregulation of the differentially expressed gene, and specifically of the tumor suppressor gene and oncogene, potentially through their trends. A similar pattern of immune cell expression was observed in the analysis of the GEO dataset, which further supports our findings. Remarkably, the CD4 and the CD8 T cell appear to have a twofold effect on the disease progression. While CD8 T cells are generally known for anti-tumor properties, recent results have highlighted their draw functionality, which varies depending on the tumor microenvironment and their state of exhaustion. Similarly, CD4 T cell can exhibit both anti-tumorigenic and pro-tumorigenic roles, depending on their subset differentiation and functional state. We concluded that this dual effect could have contributed to a stabilized level of progression and probably aggressiveness, contrary to many previous reports. Our findings suggest that its tumor microenvironment does not necessarily exert a purely positive or negative influence on cancer aggressiveness. Instead, the presence of immune cell fraction withdrawal functions appear to create a balancing effect on the progression of colon cancer in the hair subtype, as illustrated by the downregulation of both oncogenes and the TSGs in the same subtype. So, if the gene expression changes during the disease progression from early to late stage are not accounted for, the two subtypes could be mistakenly regarded as a single subtype, underscoring the importance of the normalization method for tracking cancer progression. We were also able to identify two pathways, which is the cell adhesion molecules and the ECM receptor interaction, which both play crucial yet opposite role in colon cancer. The downregulation of both pathways during the disease progression results in an altering effect that maintains a consistent level of aggressiveness. This further explains our findings regarding the balancing effect of the tumor microenvironment on colon cancer progression in the hair subtype. Moreover, a recent report also identified a strong correlation between the immune cell infiltration and the ECM receptor signature. This insight provides a new foundation for the future design of anti-cancer drugs, which focus on the unique molecular characteristics of the identified subtypes and tumor environment. In conclusion, our study demonstrates the existence of a complex relationship between the tumor microenvironment and gene expression in explaining the molecular heterogeneity of a novel colon cancer subtype. The simultaneous downregulation of both the CSG and the oncogenes in the hair subtype suggests the factors beyond mutational burden, such as the TME regulation mechanism, which contribute to this downregulation. In addition, the regulation of the cancer driver gene is negatively correlated with the CD4 and the C8 T cells, denoting a likely dual role of immune regulation in controlling S subtype progression, from regulation of the TSGs and the oncogenes that play opposing roles in cancer. Without studying tumor progression along stages, the distinction between the subtypes will be difficult and overlooked. The strong findings here enhance an understanding of the genetic characteristics and molecular mechanism that is involved in the heterogeneity and progression of colon cancer. Therefore, this knowledge improves colon cancer prognosis and advances the development of precise targeted therapies. Thank you.